Hi, everyone. Welcome to Foresight's Biotech and Health Extension seminar series. I'm really excited to have the Biomarkers of Aging Consortium committee and folks on again for the seminar. I think you've been joining us now a few times, and then of course, Vadim, you've been no stranger at our events, and was really it's an honor each time to host you. So thank you so much for the fantastic work that you guys are doing at the consortium. I think it's really elevating the biomarker entire study and also like dissemination of biomarker knowledge, which I think is like one of the biggest bottlenecks, really holding the broader aging community and aging field back. So thanks a lot for the work that you're doing. We're excited to hear any updates that you have to share with us, especially about the longevity study, perhaps later on, but really focusing on the on an update for now. So without further ado, Vadim, please take it away, and I'll be in the chat monitoring for questions, and I will be sharing all of your bios as you guys top on. Okay, well, Vadim, thanks a lot for joining. The stage is yours.
Thank you, Alison, for the opportunity to present. I'm listed as a speaker, but in fact, it's a group activity from the consortium, and so I will just start and then introduce other speakers. So this is the plan for the presentation today. I will just say a couple of words, and then Jesse will talk mostly about roadmap work group update and also about the challenge, a little bit about challenge, and then Madi and Chiara will talk about the longevity study, which is a new initiative that was initiated by the consortium. So just a very brief information about the group. Formed the group about 2 years ago, and this was spearheaded by Madi Mokri, who is an instructor in a lab. It was really his idea to bring this together. And now it's a really large group of people. I would just comment from my own kind of perspective that quantifying aging and longevity has been a major advance in the field. And until recently, it was not possible. Now there's a proliferation of studies in all kinds of context and model organisms, and it's literally impossible to follow. That's why we need a platform to discuss and debate and develop new approaches. And this platform is provided by the consortium. This is really the purpose of this group. And there are several activities here. So first, if you would like to learn about the group, there is information here and on the website. So here, consortium. Then Madi will talk about the longevity study. This is a new initiative currently at Brigham Women's Hospital at Harvard, and hopefully will be expanding. There's a conference that is coming actually very soon. Here is November 1st and 2nd. I just will very briefly mention about this conference. We expect 50 speakers plus 60 flash talks, which is 1-minute talk from poster presenters, plus posters, of course, and there will be all the leaders in the field, as well as representatives of like governmental organizations, funding agencies, XPRIZE, Evolution, ARPA-H, and editors and stuff like that. So it's going to be really an amazing gathering that is coming soon. So looking forward to see many of you there. Then also a biomarker challenge. This is something that we initiated about a year ago, and it's a competition for aging biomarkers where we collected data for 500 people from our Mass General Biobank, and only we know who these people are and what their conditions. Many of them passed away. And so we ask a question like, predict their age, like develop new models. Are these models better than any published models? And we already completed first phase for predicting chronological age, and the second phase winners will be announced at the conference very soon, next week actually. And this will be a prediction of mortality, the time to death, basically. And BioLearn is another tool that the group developed to harmonize the data and really to use various biomarkers of aging and clocks. With this, I will stop and to give more time for other presenters we will not cover everything, all the activities of the consortium, but we will cover 2 particular topics here today. Thank you very much.
Thank you, Vadim. So I'll pick up from there. My name is Jesse Boganek. And I want to tell you about what we've been working on with the roadmap work group. So this is, I would say, the longest standing program of the consortium. This is the kind of hub of our scholarly activity. And it's called the Roadmap Workgroup because the point is to try to define a roadmap to bringing biomarkers of aging to the clinic where we hope they're going to be useful. And I just want to start by thanking the Roadmap Workgroup. You see all the names of the members up here, and I'm sure you recognize all of them. These are really leaders in the aging field across the world who we're very, very privileged to work with to do this work. So just give you a brief snapshot of the history of this group. So the idea for the consortium, as Vadim said, was really conceived by Madi, and this happened primarily through discussions at Cold Spring Harbor almost exactly 2 years ago now. And very quickly, we began the Roadmap Work Group using this Delphi-based consensus building to try to bring people together to develop some consensus around key concepts in the field. And in August of last year, we published our first collaborative manuscript in Cell. This was about classification, evaluation of aging biomarkers. And because the model was so successful, we very quickly started work on a second manuscript, which was published at the end of last year. And that was on validation of biomarkers of aging. And around the same time last year, we had our first Biomarkers of Aging Symposium at the Buck Institute. I'm sure many of you were there. And this was a very, very nice and successful inaugural symposium for us, from which now the conference this year has grown out of as an expanded program. And one of the things and this is a group picture after you see yourself in there. And as Vadim already mentioned, our conference this year is coming up just next week. So we hope we'll see a lot of people there as well. But one of the things we did in the background of the symposium last year was that we distributed a survey to our panelists and speakers. This is just a snapshot of what it looked like. But we asked not just basic demographic questions like you see here, but really to get an idea of the key challenges that are inhibiting the progression of biomarkers of aging to the clinic. And from this, we prepared a third collaborative manuscript, which was published a few months ago. And the topic of this manuscript was challenges and recommendations for the translation of biomarkers of aging. QR code is here if you'd like to read it. It's published now. And the topic of this paper is exactly what the title says. The key challenges based on about 40 responses that we got. We synthesized them and we summarized these 6 key themes that emerged over and over again. And this went from data sharing for biomarker development and validation, which is a huge problem, or a challenge, I should say, that we face, relative importance of various criteria for biomarkers, the age range at which they should be applied, the minimal criteria that we need to reach to kind of legitimize these for clinical use. How can we position biomarkers of aging in the current status quo of healthcare? And last and most importantly, and what will lead into what Madi and Chiara will tell you about today, is how do we connect biomarkers of aging with healthcare outcomes and give, therefore, clinicians actionable insights so that they can actually do something with the data that they get from these biomarkers of aging. And ultimately, the hope is to progress to use them in more preventative settings rather than reactive settings. So the current status quo, as I'm sure everybody here knows, is that we can measure biomarkers reasonably well. And of course, the evaluation and benchmarking, innovation and making new models, these are other programs that the consortium is spearheading through things, for instance, like the Challenge series, as Vadim mentioned. But the problem is we produce these numbers that we believe to be representative in some way of biological age. But the question that clinicians often ask is, well, what do they do with this information? And this lack of an answer to this actionability ultimately lowers the translational utility at this point. But surmounting this challenge has the potential to be able to give us these clear interventions and insights. For instance, people might need more frequent screening if they get an aberrant result. They may need to change the therapy, or they may need to be put on a targeted therapy. So ultimately, this gets us to higher translational utility. But one of the key challenges that underlies this conceptual challenge is, of course, how do you get to this point where you're linking biomarkers to outcomes and insight? And this is actually very difficult. So one part of the challenge is that there are very few good data sets that allow you to do this, where you have both biomarker data and outcome data. And the other challenge is that there are really very few studies that were purpose-built to do this in the context of aging and longevity. And so that's why very recently we conceived this idea for a new prospective cohort collection of a deeply profiled longitudinal study. And this is called the longevity study. And with that, I'm going to hand it over to Madi and Chiara, who are going to tell you all about that study.
Thank you, Jesse. Let me share my screen. Share screen. Share slideshow. Do you see my screen there? It's in the full screen mode. Okay. If it's timed. Okay, hopefully it's not timed. I want to start by thanking Allison and Forsyth for hosting us again. And I'm a junior faculty and instructor at Harvard Medical, working at the Gladyshev lab. And just to make a quick disclaimer, these are my affiliations and other roles. The one that relates mostly to this talk is the Biomarkers of Aging Consortium, which I'm co-directing, and a correction to Vadim introduction that it's spearheaded by all of us, by Jesse and Chiara, and who joined me on day one and stuck with me the whole time, and the great support from the Methuselah Foundation and Foresight with Allison and everyone else. But today, what we want to spend some time on is the biomarkers in the context of human interventions and how biobankings are enabling us to validate these biomarkers. So I think everyone here agrees that there's a huge potential for these omic biomarkers, especially the new generation of biomarkers, for application in longevity interventions, starting from monitoring for healthy aging to stratifications, or after validation, hopefully using these biomarkers for assessing longevity interventions. But we also, most of us agree that we need to do this validation. We need to better link these biomarkers to functional outcomes and diseases of aging, and understand which one of them are causal, which one of them are upstreams, and which population they apply to. And just to give you a few examples of each one of these, recent examples that we or our networks, our consortium members have been involved on the healthy aging and association studies. I think we have many studies that there's no doubt that these biomarkers from any domain, epigenetics, transcriptomics, metabolomics, and proteomics, are associated with age-related outcomes, with diverse age-related outcomes. And you can see our latest review published in Nature Medicine last year with the help of the large group of experts. And we show that the studies are now validated, at least for association in diverse population, many of them. And on the stratification side, you can refer to the work by Andrea Meyer, our advisor, our steering committee member. And now they are using these kind of epigenetic biomarkers, some of these epigenetic biomarkers, to stratify patients and assign protective interventions to these people. We still need to wait and see how they perform, but we know they are already being used for stratification in trials. And on interventions, I'll just give you an example from our network. There's this new program that's going to start soon. It's called FAST from AFAR, led by Dr. Belsky and our own Nir Barzilai. You can learn more about it on AFAR website. But these are all just great initiatives that shows the promise of omics biomarkers in the context of longevity interventions. One specific advance that enables a lot of these new discoveries and new validation efforts are biobankings, our largest scale omic, multi-omic biobanking. And here I want to talk about two of them in particular that we've been using more and more recently. One of them is the Harvard Mass General Biobank, Mass General Brigham Biobank. And right now they have almost 150,000 patients, or relatively or some healthy population as well. And for a lot of them, they have omics and medical records, full medical records. And we use this data to host, to generate omics, additional omics for our biomarkers of aging challenge, phase 1 and phase 2. And then you can learn more about it on our website. But I just wanted to highlight how useful these biobanks and these omic samples are. We've been also using them in our recent research to show that you can improve your association, your examining of age-related activities by age-related outcome by combining different omics from the same individuals. And these are two preprints that are publicly available. And the other one that I wanted to mention is the IPOP cohort. It's a completely different design. It's a smaller cohort of 100 plus participants, relatively healthy, but they've been studied longitudinally now for more than 10 years and deeply profiled, not just omics, but a lot of other assays. And it enabled multiple individual level findings, including early cancer detections from people that we know. I joined this study 5 years ago at Stanford, and it also enabled some of our research, including our new work on studying more than 1,000 lipid species that came out last year in Nature Metabolism. And there are a lot of other good cohorts and biobanks that are generating on a weekly basis. They are generating omic data, but bear in mind that every one of them has their own unique advantages and limitations. A lot of these are collected samples years ago, and they haven't followed the most advanced practices for storage or processing. And some of them are running out of all their samples, and some of them are designed for a specific population, specific diseases, and any one of them is unique in some sense. But working with all these cohorts and reviewing all these cohorts and collaborating with the directors of a lot of these cohorts who are now members of our consortium, we've been thinking about an ideal or an optimized biobank for the longevity community for running design for aging studies, including longitudinal multi-omics and functional measures. And we've been thinking how we could enable this biobank, how we can create that and pilot this biobank, and which kind of studies it could empower us to do. So on the omics side, so far we think epigenetics, transcriptomics, proteomics, and metabolomics are some of the key that our network at least have been validating them. And we want to see if they could be used to predict or to start with to predict age-associated outcomes and functional decline. And then if they are causal to these declines, to these age-related declines and diseases. And the other part that we want to work on is how we can make this data more available with open consent, with broader consent, with easier data access, which came out as one of the main challenges with working with omics data for this kind of a study. And we spend a lot of time just to show you one example of these functional assays so omics have been very well validated in our studies and our network, but just to show you the other side of it on the functional side, there are also now good measures like VO2 max that have been very well validated across populations. And the interesting part is we can see the decline in this measure starting from 20 years old. So now we have, and they associate with diverse age-associated outcomes, age-related outcomes, decrease in including cardiovascular diseases, diabetes, stroke, and lifestyle. So it's very broad, and these are good measures. It's more difficult than the traditional functional assays like grip or walking distance or balance, but they are becoming well validated on the functional aspect of the age-related measurement. And so putting all this together, we proposed a longevity study, a longitudinal longevity study. And this year we've been spending a lot of time pre-piloting with our N of 1 and putting together the pilots and designing the consent. I'll tell you a little bit about it. And the next week, we are starting our pilot with our first 100 participants, and it will be at the same time of the conference. So we'll invite some of the conference participants to join the study. We are not addressing all the challenges with this pilot, but we are mostly learning and building the capacity and timing everything and building the infrastructure for our launch time, which is 2025 with at least 300 participants. And the plan is to do annual study of these people for 5 years at least and see if these omics measure could be linked to the functional measurement for these people. And our N of 1, we invited an active professor of mathematics, 90 plus years old, and with his permission, I'm showing his picture here. And we tested some of our protocols, and these protocols are based on more than 10 different protocols from our groups and anything that we could find publicly from a larger cohorts. And picking anything that's relevant to study of aging or longevity and applies to young and old. So that was our criteria, and we consolidated a lot of these protocols. We worked a lot with our IRB at the hospital, at Mass General Hospital, one of the most strict IRB committees, and we approved a consolidated protocol and a broad consent that allows sharing of samples and data. And we also tested some of these protocols for sample collection and processing and storage for DNA, RNA, and plasma. And these will be all publicly available, hopefully by the time of the conference next week. And some of this information you can already find under our domain at longevity.bwh.harvard.edu. And you can also find about the approval of our pilot study at BWH. And most of these pre-pilot and these legwork have been done at the Gladyshev lab. So special thank to the Gladyshev lab. And I'll stop sharing and I'll let Chiara to tell us more about the pilot and the details of the pilot study that we want to do.
Let's hope this works. Sharing here. All right. Can everyone see my screen full? Sounds good. So yeah, thanks for the talk so far. My name is Chiara Herzog, and I'm a senior postdoc at the University of Innsbruck in Austria, where I've been kind of organizing and leading some human intervention trials with over 1,000 people at this point. So hopefully I can contribute some of my background to this new longevity study. And I wanted to give you a little bit of an overview of what the study design looks like at this point and how we're hopefully bringing these resources also to the broader community so we can start running decentralized studies. Before I start, I wanted to mention, as has already been said before, obviously this is not an effort led by any single individual, but we have a very great leadership team, great collaborators, great advisory boards, and also kind of people that are helping with the day-to-day tasks. So just a massive thank you to everyone on this slide. We're only starting out with this project, but so far it's been really great to work with everyone, and hopefully we can expand the team as the study expands as well. Great. So what are the aims of the pilot study? I guess as is kind of self-explanatory that at this point, this is kind of a trial to see how can we start such a study. So what we really want to do is establish a proof of principle. Can such a longevity study be done in such a short amount of time, which I think is worth emphasizing to anyone who has done a study. Usually this takes years, even just to get a protocol and to get IRB approval. And so far, we've managed to get approval within 3 months and really set this up. So I think this is worth mentioning. So we want to see kind of what's the feasibility, interest, and acceptance of such a study. We also obviously need to identify what are potential bottlenecks. So next year, when we start the study, we're prepared for the worst, so to say, but hopefully everything will go smoothly. And also we want to trial what are the best methods for data collection, functional tests. As mentioned already, we want to cover broad functional tests that are not only applicable in older populations, but could in principle be used to track function throughout life course. So with that being said, our timeline is relatively strict. So we're targeting around 100 participants in this pilot. And as you can see, we only have 3 days to do it. And more concisely said, this will be kind of in the context of our conference that will be happening. So we have quite a busy schedule during those days, but we're looking forward to that. And the samples will then be analyzed in early 2025. So we are planning to do multiple omics in that timeframe. As this is part of our study, this will be happening at Harvard Medical School in person. So people that will be participating will need to be kind of on site, which facilitates our sample collection and all of these things. So what kind of population are we targeting with this study? We basically want to have a very general population aged 18 or older. And we have basically been prescribed a minimum weight limit by the IRB. I think this has mostly to do with blood donation, although we're not taking a lot of blood. So we have kind of a minimum weight limit, but we also want people to basically have no diseases which could affect blood donation, such as sickle cell disease or anemia. And we want no symptoms of heart conditions in the last 6 months, just basically for safety with the blood donation. Also, to ensure that participants are, not struggling with the donation. And this could also then influence the omic biomarkers. We want the participant population to be in general good recent health, so free of cold and flu symptoms on the day of collection, and also basically within the last 2 weeks. And in addition to that, no large recent blood donation. So basically, if you've been giving a lot of blood recently, that would be an exclusion from this population. So these are very basic criteria, but in principle, the population that we're targeting with this study is quite a broad population, and we're not restricting it to anything. And we can discuss the downsides or advantages of that later, perhaps. What will the study procedure look like? So we're quite limited in time. As I said, we have only a few days, so it needs to be quite compact. So we will have information and written informed consent, obviously. And we will take blood, so a maximum of 50 milliliters, which I have been told is the approximate of 2-3 tablespoons, although this is kind of a weird picture to think about blood on a tablespoon. But anyway, so we'll be on the day, we will be processing, aliquoting, and storing these samples appropriately. And importantly, we've ensured that this is all optimized for omic measurements, because for a lot of older studies, of course, the samples were not taken with the context of omic biomarkers. So now looking forward, we can really optimize for that. We will have digital questionnaires, and I will talk a bit more about this in a second, but we basically just want to have a very broad coverage of medical history and health, as well as other longevity-related factors. And we have a maximum of 4 functional tests that, again, should be quite quick to do. And I will also be talking about these in a second. But essentially, for one participant at a time, this should be less than 30 minutes effort. And we really wanted to make sure that this is a very accessible study that isn't taking a lot of time, because this also limits how many people might participate. Madi has already mentioned this. So at this point, we're primarily targeting 4 or 5 omics that are very well established with these samples. And I think what's very exciting about this is that we're from the outset planning to measure all of these omics. A lot of studies are saying, okay, we're only doing epigenomics or we're only doing proteomics, which makes it quite challenging to compare different biomarkers in different populations. So in this case, we will have one group of participants and we will have all the omics for them, which I think is really exciting. And then, of course, because we are interested in biomarkers of aging, we will primarily use these data to evaluate biomarkers of aging and kind of see what can we do with that. But obviously, at this point, this is a pilot study, so the more exciting scientific studies will come afterwards. In terms of the questionnaire, we were really interested in developing a sort of standardized core questionnaire that could be useful not just for our study, but also for the wider community. And Madi has already mentioned this was an amalgamation from multiple studies that we tried to capture the most important features for longevity. So, of course, we're capturing general epidemiological data, medical history, and medications. And that also includes longevity interventions. So we know that there are a lot of people, especially those possibly attending our conference, that might be, doing other supplementations to improve their health. And we're capturing all of these aspects with our questionnaire. Obviously, diet is a major factor, and physical activity. So we're also capturing that, and also sleep quality and stress levels. And I think from my perspective, this is very exciting because we're making all of our resources openly available on our website. So if you're interested, have a look. As I mentioned, the idea is that in the future, other study sites could leverage these core questionnaires and possibly add on their interesting questions. But at least the standardization of questionnaires could make the data collected at different sites much more comparable, and could possibly help us to merge these data in the future to have more useful data sets. Great. For the functional tests, we actually also have our resources openly available for that. And there we will, as I mentioned, do 4 functional tests as a maximum. And these include grip strength, which is something that we'll be measuring across all participants. It's very easy to do and is actually quite predictive even in younger populations, as well as 6-meter walking speed, a balance test, and cognition, which can be done online. So all of these are quite straightforward to do and very quick, which again was a priority for us. Obviously, in the future, we're thinking about other tests. As Madi mentioned, VO2 max is a really exciting predictor, but it's much more challenging to obtain. So these are some considerations that we will have for the future. What are the risks? We set out to make it a very low-risk study for participants, so we don't anticipate any adverse effects apart from those associated with taking or giving blood. In terms of incentives, of course, there's a kind of wider benefit to the community. Hopefully, this will enable and kickstart new research into aging biomarkers. But we're also excited that we can offer participants that are not already attending our conference streaming access for our kind of open day on day 2. And also all participants will be given a $50 voucher. So there is some benefit for that as well. But hopefully, we hope that the population that would be taking part in this study would be kind of intrinsically motivated to participate in this research. Just wanted to go over some frequently asked questions that might come up, and we can, of course, discuss later. So the data will be hosted at Harvard University, and we've worked very hard to have these very secure systems that are kind of standard associated with Mass General Brigham as well. The data will be used to enable longitudinal research in the future, in particular linking them to functional changes. So obviously, with this initial pilot, we can't say for sure what will be happening, but hopefully this will really be the platform that we can then use to build upon. I think the most frequently asked question is whether participants can access their own data. And this is also something that, you know, if I'm a participant, I would love to have my own data. While we aim to share all molecular data openly, at least in a de-identified manner. At this point, we cannot guarantee that participants will be able to access their own data. However, we know that this is a really major incentive for participants to contribute to such studies. And there's a lot of behavioral research as well into kind of modulation of your behavior once you receive biomarker reports. So we are actively thinking about it and working how we could make this happen in the future. And yeah, for any questions, feel free to ask us now or head to our study website, and I'll just put the link here. And thank you for your attention.
All right. Awesome. Cool. Is there anyone else to follow to close it out, or should we move to Q&A?
No, that's it. That's all on our side. Yeah.
Cool. Great. Well, you already, of course, you have the first hands already popping up. Thank you so much. It's really awesome. I mean, I'm involved always in bits and pieces, and so it's really, really awesome to see just like the progress overall that's been happening. So that's really cool. Okay, cool. We already have a few questions. I guess, are you just ready to take them? I think we have Micah and then Tom. Oh, Micah just removed his hand. So Tom, if you want to ask first, that would be great. Micah is, I think, rejoining. If your mic is still not working when you rejoin, Micah, I can read out your question. That's no problem. And anyone else, feel free to raise your hand to ask a question, or feel free to drop it in the chat and I'm going to read it out for you. Tom, you go.
Great. So I totally understand the value of having new cohort studies that are designed from the beginning to help with longevity. But I'm also wondering whether you see opportunities to enhance existing biobanks and cohort studies. So I've been working with the UK Biobank on expanding their core funding. So we were able to get the UK government to put in £25 million, which was matched by private philanthropy and the private sector. They're also looking at measuring the relative abundance of 5,400 proteins in all 500,000 participants and doing 100,000 repeat visits. So I'm just wondering, both with respect to biobanks and cohorts that exist today and those that are in the planning stage, like Our Future Health, are there some enhancements that you would be particularly excited about from a aging and longevity point of view?
Maybe I can. Vadim, go ahead.
Go ahead. Go ahead. Yeah.
If Vadim, you want to talk about UKB and some of our more than 2 years working with them, go ahead.
So let me start, and then maybe Madi, you can continue. So we actually work with UKB very extensively. We have many projects in the lab that leverage the data. This is amazing data set. I think the best phenotyped cohort, and there's lots of omics, and I'm really happy that it will be expanded with the additional proteomics data, because the existing proteomics data set for 53,000 people is already amazing. It's going to be 10 times larger. It's going to be the best ever. So, but having said that, there are also many challenges. And one challenge is like data availability. It's really a nightmare to get the data. And the more we study, actually, it makes more complex, more complicated. We want to set up a precedent of open access data, something that maybe would be followed by other people. It's not just the value of the study itself, because it's going to be relatively small, but they set an example and really support the community. Whereas, of course, we want to help with other cohorts as well. Okay, Madi.
Yeah, no, you said it exactly. We've been working with them, but for us, it's about addressing some of the existing challenges. So I see one person asking so UKB doesn't return the result back to the participants. So we've been spending a lot of time with the IRB looking into what are the challenges. So there was a question, what stops you from giving the results back to the participants? It's not simple. So we can have a separate session just discuss that specific issue. But now we are working on those by running these pilots, by running these smaller scale study, addressing some of these challenges. How can we return back the omic results back to the participants? How can we translate them, interpret them? How can we make it useful for the participants? So that's one of the differences between UKB and our MGB and the studies that we are doing, and many other open questions that we are trying to address with the study.
All right, awesome. We have lots of questions in the row. Micah, why don't you ask your next one? I'm not sure if Madi just answered it, but why don't you take it a shot?
I can pivot to a different question. Have you guys considered doing data gathering and these studies in a jurisdiction that's a little more friendly to open access data, somewhere that doesn't have HIPAA and whatnot?
Maybe I can give you a bit of information about what we are doing with other cohorts. One of the main goal of this study is to set it up as a model for other institutes and other clinics, other entities to follow. And we have that in mind every time we are designing a protocol, a functional test, an omic. So taking into account the budget, the time, and the availability, accessibility of those assays. And the other thing is we are publishing not just protocol, but all the consent information, all the language that we use, all the detail about these studies, including references, including the best practices. Everything that we develop will make it publicly available to the community so that other entities can replicate or can follow. And with that, if anyone from other countries, other entities wants to collaborate with us, we can help them. We can work with them and understand also the challenges that doesn't apply to us. For example, we are looking into challenges that apply to Asian countries or Gulf countries or African countries, and how can we make these studies relevant to them as well?
Awesome, great. Next one we have Lucas Chu.
Hi, Lucas from Harvard here. Question: do you have any hypotheses that you're testing in particular? For instance, are there like biomarkers that you suspect are the most important, predictive, and like how else did you come up with the ideas?
Yara?
Sorry. Yeah, no, that's a great question. So in this stage of the pilot, because we are limited and we essentially only have a cross-sectional data set, there's not that much we can do yet, but we're really trying to learn. In terms of additional hypotheses, yes, of course, this is something that we are setting up, and we're working on a few different study designs at the moment to kind of see what would be the most useful for the cohort and then optimize kind of the sample number and design towards that end. We are also, and I think this overlaps a little bit with our challenge, that we are on that side also already working on which biomarkers, for instance, are the most predictive for mortality and these sorts of things. I think biomarkers that pop up in the challenge would be quite interesting to then look at in the longitudinal study, in particular, because most studies so far are not longitudinal. And actually, there's very limited information on these kind of dynamics. So I think that would be quite complementary, but yeah, it's still a little bit of a process to get there.
Anything
To
you can
add
share
to that-
on hypotheses?
Yeah.
or biomarkers?
So to add to that, on the study design, two main aspects is the population and the research question. On the population, I can also share that we invited Harvard medical students, first-year students, to join us so that we can longitudinally follow them every year with these omic biomarkers and functional assays. So that could enable some kind of unique longitudinal omic functional studies in younger population. And hopefully that will be complemented with VO2 max studies and more complex studies that we can take more time to do them locally. But for conference participants, it's a different setting. They'll be here only for 2 days, so it's a different set of limitations.
Awesome. Next one, we have David, then Michael, and then Jim.
Hi, as the program goes on. Oh, hello. Hi, Madi. The data that results from it, the interventions, will provide insight. Will the protocol be changing along the way in response to those insights? We may find things that are terrible that we think are good today, and so forth. So is there a plan on how to address that without fouling the data and the study itself? Thank you.
Thank you, Dave, and thanks for supporting our studies. Maybe Jesse can take on that one with his clinical experience, translational experience.
Yeah, so it's a very good question, and of course, kind of trying to predict the future a bit, right? But actually, I think that one of the most instructive things that's going to help us specifically to deal with this issue is Nir Barzilai and Dan Belsky's new FAST initiative, where they're going to identify biomarkers that are changing in response in kind of existing clinical trials. So that may be a way that will allow us in parallel to very quickly understand which of these biomarkers or types of data that we will be collecting in the longevity study may be impacted by various interventions. And as I think has been emphasized many times now, right now we are in a pilot phase, potentially even a pilot of a pilot phase. I think things will be very dynamic as time develops and as we learn. So of course we will be adapting to the latest science as the years go on.
Great. Could I follow up, Allison?
Oh, we still have 9 minutes. Go for it.
Yeah. So we just opened a longevity clinic in Tysons Corner, Virginia, and we have the VO2 max and pretty much everything that would be needed in order to track individuals. So that could be a collaboration that would extend the ability of the study to, keep track of folks without having them go to Boston or whatever it is your current plan is. Just thought I'd bring that up.
Thank you, Dave. Yeah, we are talking to clinics, to institutes. I actually talked to your clinic yesterday, and I'm planning to visit next month. So we are learning from you and all the advances that you have made.
Okay. So are you actually coming to Tysons to
the DC one.
Okay.
Oh, Michael, you go.
Hey guys, great job at getting the study together. I'm excited to follow. Yeah, how have you thought through frequency of sample collection? Obviously, there's hurdles to doing it very frequently, like weekly or monthly. But do you have any sense of if you did it more frequently, weekly or monthly, could there be a 10x improvement in sort of the resolution of the omics data for actually, inferring this stuff?
So one thing that came up during a discussion with one of the advisors that we didn't list yet was using apps and digital biomarkers that we haven't included in our protocols yet because it's just something more recent and we have less protocols and it has its own unique challenges with ethics and IRB. So it needs the extra time. We need the extra time to go through them. Maybe by the time that we are launching next year, we can also take care of that. But for omics, doing it anything more than a yearly, it would be very costly and challenging. Just to give you some rough number for the proteomics that we are doing, even on the pilot, it costs around $1,000 per sample, just for one time. So now you can estimate how much it would cost to do multiomics. But this is the largest panel of proteomics with 11,000 species. So I guess it's balancing the budget and the feasibility and the benefit. But yeah, digital biomarkers is one way to go.
But I guess leaving aside practicality and costs, if you could have a full omics collection, whatever, once a month, once a week, like, do you think it would be way more valuable than the annual collection, or kind of like, who knows?
Yeah, we don't have the answer to that because very few cohorts have done that. I can refer you to one study by Mike Snyder group that we collected his blood every month for 2 years, and we looked at his biomarkers. And there's a paper in one of the Nature journals on this data, and the data is also publicly available. But it was a sample of one, and it cost more than $100,000. So we don't have the answers to those questions yet.
Maybe I can
fair enough.
add a little bit of context. So we've actually done a study in like 150 people where we did epigenetics and microbiome and all of these every 2 months. And we can say that some biomarkers change much more rapidly than others, which makes complete sense. So some epigenetic biomarkers change faster, the microbiome change much faster, metabolomics, like almost no similarity between the 2 time points. So it really depends what you're measuring. I think it would be super interesting, but I think at this point, Madi has a good point that we need to balance the practicality with what we can do, but hopefully somebody will be able to complement these data.
Yeah, maybe
Cool. That'll make sense. Thanks.
I will also add that, well, if there's a funding available or somebody is willing to support, actually, it would be, of course, very good. So we have a project in a lab, as an example, where we try to relate biomarkers at different time scales, all the way to like what happens during the day, like the hourly kind of changes. And this also would be very useful to do it. But again, so it's always a compromise between the resource availability and outcomes that you want to measure.
Makes sense. Thanks.
Awesome. Well, maybe I will read our Jim's question and then we go to you, Nicola. It's, as you know, many of us have been advocating that Congress give NIH money to fund research on biomarkers of aging. If NIH suddenly had 150 million per year for this, could you describe what the larger scale research would look like?
We have one of the advisors, Nir Barzilai, who's been looking for that money. So maybe Nir can comment on that. Nir, you're unmuted.
I think Nir's just trying to say that if it was up to him, he'd give the money to the longevity study.
Of course. Well, Nir, if you don't want us to put words in your mouth, feel free to
Sorry,
update.
no. Sorry, I had other documents and it didn't get the. So how can I help? I'm sorry. What do you want me to say about funding?
Oh,
If we
basically
$150 million a year through NIH, what would the scaled up?
No, I look, I think I'm in New York now, and we don't do prediction in particular about the future, right? We just learned that radical life extension is probably not going to happen in our life. But I would just say that the breakthroughs sometimes are through organizations like you or like Tom Khalil is having and many others. And the organization that I think is going to be the most proactive in this is ARPA-H, right? And this FAST initiative is something that ARPA-H is very interested. So I'm very careful. They're excited, but we're in the progress with those guys, all, with Madi and Chiara and Vadim and everybody in the process of making a good application. But I think for me, the breakthrough is that I think we'll have a lot of biomarkers and there's a lot of money going to biomarkers. But at the end of the day, we want to have a biomarker that if we do intervention versus whether it's exercise or diet or sleep or social connectivity, all have biological input, or drugs, most important drugs, that we can actually show that we're getting younger, right, with age. That is the major thing. A lot of the biomarkers we have now are not changing, okay? Not all the epigenetic biomarkers are changing when we do intervention. So I think we can find the biomarkers of aging and those that are changing, and I think this will amplify things on their own, because individuals, just like as they want to see their cholesterol or their hemoglobin A1C levels, they'd want to see progress, and this will get more money, more commercial companies into, and this will be good for the field.
Cool. Next one we have Nicola. Thank you, Nir.
Okay, so we don't have much time, but I really want to raise the question about the right hands of the equation, because I think it was a great idea when we moved from making clocks that predict your age to making clocks that predict when you'll die. Still, death is not the best possible outcome. Just before death, there's a very fast decline of many things, and things are dwindling out. That's not necessarily what will help us keep good health. And so my question is, if you guys are thinking about other outcomes and what the better outcomes are, and we can call that a measure of health, but what kind of phenotypes we can use to analyze the data?
We are at time, so if anyone wants to do a 10-second answer to this, that would be great.
That's an open question.
The answer is
Vadim,
not
go ahead.
known. The answer is not known. That's one of the reasons we have this consortium, just to debate and discuss these ideas, because one can build biomarkers based on chronological age, based on time to death, based on functional changes, disease occurrence, and so on. But we don't know what's the best. What's the best biomarker that we could quantify aging, whatever aging is, and target it? So we need more studies on that.
And I would argue that neither death nor age is a good phenotype. And I agree with you that we have to find appropriate phenotypes that measure health.
The good news is they are also getting better. The example of VO2 max is just one example.
Well, we're going to cut it down. I know that there's plenty of questions. Some people are just starting, but thank you so much for joining us. I hope that we get the pleasure of having you back and again in a few months. Given how fast you're moving, I'm sure that you have a lot of other updates to share then. But yeah, I'm really excited to see all of you in Boston very soon for the conference, and many of you probably on this call as well. So it would be good to meet up in person again. Thank you so much, everyone. And yeah, I will see you all very soon. Have a good one.